My topic is Entamoeba Histolytica. The word amoeba is derived from the Greek word meaning change. Amoebae are structurally similar to protozoans which have no fixed shape. Amoebae are classified as either free-living or intestinal amoebae. Many amoebae inhabit the human gastrointestinal tract such as Entamoeba fragilis, Entamoeba histolytica, Entamoeba coli, Entamoeba dispar, Entamoeba gingivalis, Entamoeba hartmanni, Entamoeba moskovski, Entamoeba pollici, Chilomastix mesnili, Endolimax nana, and Iodamoeba butchleri. Free living does not cause infection, but several types of free living may cause infection. The parasitic amoebae inhibit the alimentary canal. All intestinal amoebae are non-pathogenic, except Entamoeba histolytica. Entamoeba histolytica is a common protozoan parasite found in the large intestine of humans. The parasite is responsible for amoebic dysentery and liver abscess. It is the third leading parasite cause of death in developing countries. Entamoeba histolytica was discovered by Lost in 1875, who demonstrated the parasite in the dysenteric fecus of patients. In 1890, William Osler reported the case of a young man with dysentery who later died of a liver abscess. The most common source of infection is fecus oral root and ingestion cyst contaminated food or water by human fecus. Morphology. Entamoeba histolytica occurs in three forms: trophozoite, precyst, cyst. Trophozoite is the vegetative or growing stage of the parasite. It is the only form of present in tissue. It is regular in shape and varies in size from 12 to 60 micrometer, average being 20 micrometer. It is large and actively motile in freshly passed dysenteric stool, while smaller in convalescents and carriers. The parasite, as it occurs free in the lumen as a common cell, is generally smaller in size, called minute form. Cytoplasm. Cytoplasm is divided into two portions: a clear transparent ectoplasm and a granular endoplasm. Ingested RBCs, tissue granules and food materials are also found in the endoplasm. Nucleus. It is single, spherical in shape and size ranging from 4 to 6 micro. Nucleus contains central karyosome and fine peripheral chromatin. The nucleus is not seen in the living trophozoites but can be demonstrated in preparations stained with iron hematoxylin. Pseudopodia. It's a finger-like projection formed by sudden jerky movements of ectoplasm in one direction, followed by the streaming in of the whole endoplasm. Precyst. It's the intermediate stage between trophozoite and cyst. It is smaller in size, 10 to 20 micro. It is round or slightly ovoid with a blunt pseudopodium projecting from the periphery. No RBC or food materials are found on its endoplasm. Cyst. It is an infective form of parasite. The cyst is spherical in shape, about 10 to 20 micrometer in size. The early cyst contains a single nucleus and two other structures. One is a mass of glycogen and second one is 1 to 4 chromatoid bodies or chromatidyl bars. When the cyst matures, the glycogen mass and chloridal bars disappear and the nucleus undergoes divisions to form 2 and then 4 nuclei. A mature cyst is quadrinucleated. The cyst wall is a highly refractile membrane which makes it highly resistant to gastric juice and unfavorable environmental conditions. A mature cyst passed out in stool from an infected patient and remained without further development in soil for a few days. First, A represents a trophozoite, B is a precystic stage, and C is a uninucleate cyst. Then D is a binucleate cyst, and E is a mature quadrinucleate cyst. Life cycle of Entamoeba histolytica. Entamoeba histolytica passes its life cycle within one host. Infected form. Mature quadrinucleate cyst passed in fecus of convalescents and carriers. The cyst can remain viable under moist conditions for about 10 days. Mode of transmission. Man acquires infection by swallowing food and water contaminated with the cyst. As the cyst wall is resistant to the action of gastric juice, the cyst passes through the stomach undamaged and enter the small intestine. Exgestation. When the cyst reaches the cecum or lower part of the ileum, due to the alkaline medium, the cyst wall is damaged by trypsin, leading to exgestation. Exgestation of a cyst gives 8 trophozoites. Trophozoites are actively carried to the large intestine by peristalsis of the small intestine. Trophozoites then gain maturity and divide by binary fission. The trophozoites adhere to the mucous lining of the intestine by lectin and secrete proteolytic enzymes, which cause tissue destruction and necrosis. Parasite, when gain access to blood, migrates and causes extra-intestinal diseases. When a load of trophozoites increases, some of the trophozoites stop multiplying and revert to cyst form by the process of encystation. These cysts are released in fecus, completing the life cycle. The entire life cycle is thus completed in one host. In most cases, Entamoeba histolytica remains as a commensal in the large intestine without causing any ill effects. Such persons become carriers or asymptomatic cyst passes and are responsible for the maintenance and spread of infection in the community. The life cycle of amoebiasis. Cyst and trophozoites passed in fecus, mature cyst ingested. Then third step, exgestation and trophozoite multiplication, then exist host. Pathogenesis and clinical features. Entamoeba histolytica causes intestinal and extraintestinal amoebiasis. The incubation period is highly variable. On average, it ranges from 4 days to 4 months. Amoebiasis can present in different forms and degrees of variety depending on the organ affected and the extent of damage caused. Intestinal amoebiasis. The parasites express a large number of virulence factors including lectin, lytic peptide, cystine, proteinases and phospholipase. Exgestation of cyst in intestine releases 4 trophozoites which is then colonizes the large intestine. The binding of trophozoites with the colonic epithelium is a dynamic process in pathogenesis. After adherence, trophozoite lies the target cell by its ionophore-like protein that causes leakage of ions from the cytoplasm. The proteolytic enzymes secreted by the amoeba cause disease destruction, giving flask-shaped amoebic ulcer, which is a typical feature of intestinal amoebiasis. Trophozoites penetrate the columnar epithelium of mucosa, causing lysis and move deep inside till they reach the submucosal layer and multiply rapidly. Ultimately, amoeba destroy a considerable area of the submucosa, leading to an abscess formation which breaks down to form an ulcer. The ulcer is flask-shaped with a narrow neck and broad base. The ulcer may be localized in the ileococcal region or generalized throughout the large intestine. Extraintestinal amoebiasis. Amoebic liver abscess. In about 5 to 10% of persons with intestinal amoebiasis, liver abscesses may be ensured. The center of the abscess contains thick chocolate brown pus, which is liquefied necrotic liver tissue. It is bacteriologically sterile and free of ammonia. Liver abscess may be multiple, usually located in the upper right lobe of the liver. Cardinal signs of amoebic liver abscess or painful hepatomegaly. Fever is present in most cases. Anorexia, nausea, weight loss and fatigue may also be present. Jaundice develops only when lesions are multiple or when they press on the biliary tract. Amoebic liver abscess is 10 times more frequent in adults than in children and 3 times more frequent in males than in female. Pulmonary amoebiasis, very rarely primary amoebiasis of the lung, may occur by direct hematogenous spread from the colon by passing the liver. Hepatobronchial fistula usually results with expectoration of chocolate brown sputum. The patient presents with severe phlerotic chest pain, dyspnea and non-protective cough. Metastatic amoebiasis, involvement of distant organs is by hematogenous spread and through lymphatics. Abscesses in the kidney, brain, spleen and adrenal have been noticed. Spread to the brain leads to severe destruction of brain tissue. Cutaneous amoebiasis, it occurs by direct extension around the anus, colostomy site. Extensive gangrenous destruction of the skin occurs. Genitourinary amoebiasis, it causes lesions in the male and female genital tract and urinary tract such as penis, anus, vulva, cervix and vagina. It spreads from male to female during intercourse. Lab diagnosis, specimen, stool, pus or liver abscesses, sputum and biopsy samples. Stool microscopy. In amoebic dysentery, stool is an offensive, semi-solid, dark brown color and acidic in nature mixed with blood, mucus and fecal materials. Microscopy. Normal saline preparation of fresh fecal material reveals trophozoites with RBCs in its cytoplasm and its amoebic motility. Stool antigen detection. ELISA to detect 170 kilodalton lectin of Entamoeba Histolytica. Stool culture. Robinson medium and NH polygenic culture medium are used to culture Entamoeba Histolytica. Serology. IHA, IFA, etc. are used to detect antibodies in serum against Entamoeba Histolytica. PCR, it is a sensitive test used to differentiate Entamoeba Histolytica from other Entamoeba species. Radiological findings, X-ray, MRI, CT scan, ultrasonography for extraintestinal amoebiasis. Blood test, blood count, liver function test, kidney function test. Treatment, metronidazole and related compounds like rinidazole and imidazole act on both luminal and tissue amoebiasis. Dilocyanide, furoate, iodoquinol, paramomycin, tetracycline, imidine, chloroquine, effective in systemic infection. Oral rehydration and electrolyte replacement should be done whenever necessary. Prophylaxis, general prophylaxis is for all fecal oral infections. Food and water have to be protected from contamination with human excreta. Detection and treatment of carriers and their exclusion from food handling occupations will help in limiting the spread of infection. Health education and inclusion of healthy personal habits help in control. Thank you.